Chapter 5, An Introduction to Carbohydrates. In this chapter, you will learn about the role of carbohydrates and the role they play in life, and how they are linked together. What we'll do is we'll first examine monosaccharides, and then we'll take a look to see how they're linked together to form polysaccharides or polymers. Then we'll ask, what major roles do carbohydrates play? And then from there, we'll look at cell structure, cell identity, and energy storage. Carbohydrates play an important role in cell structure, cell identity, and energy storage, and we can have them in different forms as monosaccharides, oligosaccharides, and polysaccharides. I'm going to be honest with you, I hardly ever hear the word oligosaccharides, but I definitely do hear monosaccharides and polysaccharides. So sugars are our third important macromolecule, and sugars are made out of monosaccharides. Now, in your book, it doesn't come right out and say it, but the basic or default monosaccharide is glucose, C6H12O6. And if you string a lot of glucoses together, you'll get a polysaccharide or a very large polymer, our third macromolecule, sugars. So carbohydrates have a molecular formula of CH2O. Then outside of the parentheses you see N, and that N indicates the length of the chain. So however many number N's you have there, that will be the length of the chain. But the important thing to know is when you look at CH2O, that means that all sugars have twice as many hydrogens as they do carbons and oxygens. Does everybody agree? So for glucose, C6H12O6, notice that the number of carbons and oxygens is 6, but there's twice as many hydrogens, therefore there's 12. Carbohydrates also contain a carbonyl group, C double bond O, hydroxyl groups, OHs, and a lot of carbon-hydrogen bonds. Now, we can think that all CH2Os are carbohydrates, but they're not. Some of them can be other molecules such as formaldehyde. Sugars are important in various ways, but one of the most important functions of sugars is to provide energy. And they also serve as building blocks for larger compounds, and we'll see this later on when we talk about cellular respiration. Monosaccharides played an important role in chemical evolution. So remember that even when we talk about nucleic acids, nucleotides, part of a nucleotide has a 5-carbon sugar, and that could be ribose or deoxyribose. What distinguishes one monosaccharide from another? So keep in mind, when we say monosaccharide, we're referring to a building block or a monomer, and these are simple sugars. And usually when we talk about simple sugars, these are the ones that sort of taste sweet. These are the ones, in fact, I hate to admit it, but I'm addicted to. Okay, so how can we distinguish one from another? One, it's the location of the carbonyl group. Is it in the end or is it in the middle? Is it an aldose or a ketose? Or how many carbons does it have? For example, in nucleic acids, we have a 5-carbon sugar, which could be called a pentose, but glucose is C6H12O6. That is a hexose. So we can tell carbon monosaccharides apart by the number of carbons they have. We can also distinguish them by their spatial arrangement of their atoms. So it's very important to know where their OH and H groups are. Are they above or below the carbon plane? And we'll take a look at some figures to show you. And then lastly is a sugar in a linear or a ring form. So this is another way to distinguish one monosaccharide from another. And each of these structural changes have functional consequences. So here in this example, figure 5.1, you can see an aldose on your left-hand side and a ketose on the other. Notice notice where the carbonyl group is located. In an aldose, it's at one end, and for a ketose, it's found in the middle. Now let's take a look at figure 5.2. Here we notice that there's actually a movement of the hydroxyl group OH. So let's see if we can see that. So here the hydroxyl group is located above the carbon plane. So notice this carbon backbone. So here we see that the OH is above the carbon, but if we look at galactose, which also has 6 carbons, the only difference is on the 4th carbon, the hydroxyl group is located below the carbon plane. In figure 5.3, we see a comparison of a linear form on the left-hand side, and then on the right-hand side we have 2 ring forms, alpha glucose and beta glucose. And what's the difference between the two? Again, it's the location of the OH or hydroxyl group in reference to the carbon. For alpha glucose, it's located below the carbon, but for beta glucose, it's located above the carbon. Distinct monosaccharides exist because so many aspects of their structure are variable. So remember, aldose versus ketose, or the number of carbons, or the arrangement of OH or hydroxyl groups, or linear versus ring forms. So each of these affects the function of the sugar. Polysaccharides or complex carbohydrates are polymers of monosaccharides. In other words, how do we get this huge macromolecule? It's linking one building block to another. It's the same story as we saw for proteins and for nucleic acids. All of these macromolecules are built by a specific unit, and they're held together by a specific bond. And as you realize now, you have to know the monomer, and you have to know the bond that links each of these monomers together. So for sugars or polysaccharides, the covalent bond that links one monomer to another is called a glycosidic linkage. This glycosidic linkage, and it's formed by a condensation reaction, and it's broken by hydrolysis reactions. Glycosidic linkages can form between any 2 hydroxyl groups. So remember, hydroxyl are OH, OH. So you can have 2 types of glycosidic linkages, an alpha 1,4 glycosidic linkage or a beta 1,4 glycosidic linkage. So it says here that both linkages are between a C1 and C4 of different molecules. So we'll see in the upcoming figure. So let's take a look at this. If we look on the left-hand side, we have 2 alpha glucoses. So we have one alpha glucose and another, and we see 2 hydroxyl groups up at the top. And so through a condensation reaction, water comes out, and when water comes out, a glycosidic linkage is formed. And this is why it's called an alpha 1,4 glycosidic linkage. Okay, so now let's take a look at the right-hand side. Here we have a beta glycosidic linkage. So what's the difference between the two? Here we have beta galactose and beta glucose. So notice here we do have 2 hydroxyl groups like we had before, but one is above the carbon plane and one is below. So through a condensation reaction, water is released, but when a glycosidic linkage is formed, notice that oxygen is above the carbon planes. So this is called a beta 1,4 glycosidic linkage. And unfortunately, you do need to know the two. An alpha 1,4 glycosidic linkage is very easy to break. On the other hand, a beta 1,4 glycosidic linkage is very strong and very difficult to break. Okay, so let's take a look at some different types of carbohydrates or polysaccharides. So one of the first ones is starch. And starch is a storage polysaccharide that is found in plants. So starches come from plant material. Like I'm thinking of like potato starch, corn starch. So sugar is stored as starch in plants. And these starches are composed of alpha glucose monomers, or the building blocks. So one type of sugar is amylose. How do I know that it's a sugar? Because sugars end in ose, like glucose, ribose, hexose. All of those end in O, so that's how I know it's a sugar. And amylose has alpha 1,4-glycosidic linkages. Amylopectin is a branch starch, and it has alpha 1,6-glycosidic linkages. And we'll see that in just a second in a diagram. And branches occur about once in every 30 monomers. So here we have an example of where actually starches come from, and these look like sweet potatoes. So yum. I love starches. Alpha 1,4-glycosidic linkages. They're easily broken. So what is glycogen? Glycogen is also a polysaccharide that has alpha 1,4-glycosidic linkages, but instead of being found in plants, it's found in animals. And it can be stored in the liver, or it can be stored in muscle cells. And that makes sense, right? Don't muscle cells need energy all the time to be used? So it's stored there. Someone else also told me that a lot of Americans have fatty livers. And I think a lot of that has to do with the storage of glycogen in the liver. And glycogen can be broken down into glucose very readily for energy. And of course that makes sense. That's why it's found next to muscle tissue. So it's highly branched alpha glucose polymers, and it's nearly identical to starch. But there is some branching, and it's 1 out of 10 monomers. Then we have cellulose, and notice it ends in O, so we know it's a sugar. And this is also a polysaccharide. But instead of having alpha 1,4-glycosidic linkages, it has beta 1,4-glycosidic linkages. So it means that every other glucose is flipped. It generates a linear molecule rather than a helix, and it permits hydrogen bonds to form between adjacent parallel strands. So a beta 1,4-glycosidic linkage is very strong, and very few organisms can actually break that linkage. Okay, so let's go back to the idea of cellulose before I talk about chitin. So where do you find cellulose? Cellulose is wood itself. Don't we use wood to build our homes? Wood for furniture? The reason why we use it as a building material is because very few organisms can actually break down the beta 1,4-glycosidic linkage. So now let's talk about chitin. Chitin is also a very structural polymer, and this is found in the cell walls of fungi and the exoskeleton of insects and crustaceans. I always say that this is what gives the crunch when you're stepping on a cockroach and killing it. So its exoskeleton is actually made out of a sugar, but this sugar is so strong because it has beta 1,4-glycosidic linkages. These beta 1,4-glycosidic linkages vary with every other monomer flip. So it's that same flipping structure that we saw for cellulose. And here we see an exoskeleton, and this looks like a cicada. So that exoskeleton that's being left on the branch, which is made of cellulose, they both have beta 1,4-glycosidic linkages. Okay, peptidoglycan. And I say peptidoglycan. A lot of times maybe I'm mispronouncing it. It's peptidoglycan. But this is a structural polymer, another polysaccharide, and this is found in the cell walls of bacteria. Have you ever wondered why, when you're taking an antibiotic, that you have to take it over a course of a week? Why can't you just take one pill? Because most antibiotics interfere with cell wall formation. So cell walls have to be formed with new bacterial cells. So that's why you have to take it over a course of time. And so this destabilizes and breaks down the cell walls, and then the antibiotics, and then leaves the bacteria unprotected. So peptidoglycan, this is another structural polysaccharide or polymer that makes the cell walls of bacteria, and it's and it's made with beta 1,4-glycosidic linkages. I love the table, and the diagram is much better in your book than projected on this slide, but it gives an overview of everything that we discussed. The first two, starch and glycogen, are formed with alpha 1,4-glycosidic linkages, but then all of the others, cellulose, chitin, and peptidoglycan, are formed using beta 1,4-glycosidic linkages. And notice it looks strong, like strong metal fencing, very strong material that is used to protect cells. Okay, so the question is what do carbohydrates do? What is their function? What is their role? Well, we've mentioned before that they serve as precursors to other molecules, and that material can be used to form nucleotides and amino acids. They also provide fibrous structural material, and they can be used for cell identity and also for chemical energy. And the one I relate to the most, is chemical energy. I always used to tell myself I need to eat candy bars, especially before I lecture before class, because I need energy. I'm so animated that I need the energy, and that energy is stored in the bonds of glucose. So we know that carbohydrates can provide structural support, and it's those polysaccharides that contain beta 1,4-glycosidic linkages. So that would be cellulose, chitin, and peptidoglycan. And these guys can form sheets and strong connections, and they're very hard to break. So you know what I always think about? I always think about that cow or cattle that's in a pasture. They're chewing and chewing and chewing because they're mechanically and chemically trying to break down the beta 1,4-glycosidic linkages, but in the end, it's that particular bacteria or fungus in the gut that is helping them break it down. Remember, even on our own human diet, we have to have a lot of fiber, but that fiber is actually made of carbohydrates, beta 1,4-glycosidic linkages. Carbohydrates are also used for cell identity. So our cell membranes, on the outside of our cell membranes, we have glycoproteins and glycolipids. So if glucose is the basic monomer for sugars, then when you look at the prefix glyco, it sounds a lot like gluco. Does everybody agree? So I'm hoping that you'll think, when you see the word glyco, that you'll think of sugar. Glycoproteins, sugar proteins, glycolipids, sugar lipids, and these help for cell identity. So they can either do cell-to-cell recognition or cell-to-cell signaling. So what is cell-to-cell recognition? In a multicellular organism like ourselves, have you ever asked yourself, how do liver cells stay with liver cells and eyeball cells stay with eyeball cells? Well, a lot of it has to do with sugar. Sugars recognizing other cells that are similar. And sugar also helps to communicate between cells. So here's an example of a membrane. It's a phospholipid bilayer, and on the outside you see these like hexagons representing glucose. To me, they sort of look like little red antennae, right? And they're connected either to the phospholipid or to proteins indicated in green, or the lipids indicated in like a light blue, a silvery blue. So these are used for cell-to-cell recognition and communication, and they sort of do look like little antennae. In an experiment, what they did is they took a look at the glycoproteins located on the outside of a sperm. And this is what they found. If those sugars were removed from the proteins of a sperm, then the sperm wasn't able really to locate the egg. So here we see that cell recognition is really important, and that sugar plays an important role in this cell recognition. Okay, so where do sugars come from? Carbohydrates store and provide chemical energy. But the truth is that sugars come from photosynthesis. And we'll learn later on in a chapter just dedicated to photosynthesis that photosynthesis means light making what? Sugar. And the ultimate source of energy is from sunlight. So through plants and bacteria, radiant energy from the sun is converted into chemical energy. So in this chemical formula below, we see that CO2 and water plus sunlight actually makes sugar and oxygen. And guys, we can't live without sugar and we can't live without oxygen. So photosynthesis is one of the most spectacular formulas ever. Carbohydrates store more energy than CO2. But you know what? That makes sense to me. If chemical energy is energy that's stored in the bonds, CO2 is a rather small molecule with very few bonds. Carbohydrates are polysaccharides which have many bonds, and because they have many bonds, they hold a lot of potential energy because energy is found in the bonds. But not all bonds are the same, right? There's polar covalent bonds and nonpolar covalent bonds. So nonpolar covalent bonds have higher potential energy when electrons are shared equally. When electrons are not shared equally, they have less potential energy. In this example here, we see CO2. CO2 is not only a small molecule, but the electrons are not shared equally between oxygen and carbon. And that makes sense because remember, oxygen is very electronegative. So because of the size and because of where the electrons are, there's not much energy in CO2. On the other hand, a carbohydrate is a polysaccharide. It has many bonds. And if you look below the carbon backbone, you see all of the carbon-hydrogen bonds. The electrons are shared equally. The electrons are also shared equally in the carbon backbone. The only place where they're not shared equally is where there's an oxygen above the carbon backbone. So for this reason, sugar does supply a lot of energy, carbohydrates do. And isn't it true right before an athletic event or a marathon race, a lot of people do carbohydrate loading, and they do carbohydrate loading because they know that there's a lot of energy stored in all of those bonds in a carbohydrate. Enzymes hydrolyze energy storage, and this is how it's made available to us. So starch and glycogen are easily hydrolyzed because they have alpha glycosidic linkages, and that makes sense. We eat starches and sugars because we want that energy. So it says here glycogen is hydrolyzed by an enzyme called phosphorylase, and I know that it's an enzyme. I know that it's an enzyme because it ends in ase. So many animal cells contain phosphorylase, and this phosphorylase releases the energy in glycogen because it helps break it down to glucose. And when we have glucose, then this can help generate ATP at the cellular level. Starch is hydrolyzed by amylase, and again, we know it's an enzyme because it ends in ase. Amylase plays a key role in carbohydrate digestion. So our saliva contains a lot of amylase. If you chew on a saltine cracker and you just keep on chewing and chewing and you don't swallow, which is very difficult for me because I want to swallow, but if you keep on chewing and chewing, those amylases are going to break the starch down into mono or disaccharides. And then actually the saltine is going to begin to taste sweet. Energy is stored in glucose, and this is used to make ATP. So we'll have one chapter dedicated to cellular respiration, and in fact, this is a chemical formula of cellular respiration. What it says here is the breakdown of sugar in the presence of oxygen is going to produce ATP. It will also produce carbon dioxide and water. The energy in ATP drives a lot of reactions found within our cells, and this also helps with muscle movement. So at the end of this chapter, we have a video, and this video is accessible. If it's not in this presentation, you can go to the other presentation files and see if it works. This is the end of carbohydrates. This will be our third macromolecule, our third polymer.